Our paper deals with using expression data from single cells to reveal dynamic gene expression changes over time and to rebuild tissues in three-dimensional space. We used the mouse otocyst, the Anlage of the inner ear, as a model system. Figure 4 summarizes how we reconstructed a three-dimensional model of the otocyst, how we visualized expression information, and how we quantified the data. Shown in panel A, we used 267 of the 382 collected cells that were identified as otocyst-derived using biclustering analysis. We then performed principal component analysis, projected the data onto the first three components, and normalized the location of each single cell so that they have an equal distance to the center of the sphere, representing the three-dimensional model. Expression data of two marker genes, OC90 and GBX2, helped establishing the three major anatomical axes, which allowed us to divide the model in 6 different hemispheres: dorsal ventral, lateral medial, and anterior posterior. In panel B, we visualized expression levels of select genes for every cell. As indicated by the color bar, cells in red show high expression, yellow shows medium expression, and green represents low expression. Cells in gray do not express the gene. As a representative marker, expression of lunatic fringe is shown and demonstrates that almost all cells located on the right side of the sphere, which corresponds to the ventral domain, are positive for this marker and express it at high levels. Panel C and D introduce two different quantitation methods. In C, we quantify for each of the 6 hemispheres the percentage of cells that are positive for a given marker and the fold change differences between opposing domains. For the ventral marker lunatic fringe, for example, the majority of cells positive for this gene are positioned on the ventral side with a 7-fold higher expression compared to the dorsal half. In D, we partitioned the sphere in 8 equally sized octants. This enabled us to describe defined spatially restricted domains, such as dorsal medial posterior, which represents octant 3, for instance. For each octant, we were able to calculate the percentage of cells that express a particular gene and its associated mean expression value. Comparing the expression patterns of various genes predicted by the model with data from the literature demonstrates the accuracy of the computational representation.